Okay, good afternoon, everyone. It's my pleasure to be here today. I will be presenting our work in prioritizing causative genomic variants by integrating molecular and functional annotations from multiple biomedical ontologies. As you may all know, that the genomic variant is the DNA sequence differences among individuals, which involve the concept of polymorphisms, variations in the DNA sequence that contribute to the diversity among individuals, also play a crucial role in the development of the diseases. And there are several types of these genomic variations in the DNA sequences. These variations comes in all sizes. It range from a single base pair change known as a structural variants or SNPs in short, to much larger changes that involve over 50 base pairs known as structural variations. And these genomic variations present a challenge in diagnosing rare diseases using whole genome and whole exome sequencing studies, which are widely used for diagnosing patients with rare diseases. However, many patients remain undiagnosed, either due to the undiscovered disease gene or disease phenotypes, or the novel phenotypes that arise from a combination of variants in multiple genes, as interpreting the phenotypic consequences of the variants rely on the information about the gene function, expression, and other genomic features. So this work addresses this problem by considering the following observable as an input. First, we have the patient's genome that are represented as a list of variants, and the patient's clinical phenotypes that involve the sign and the symptoms. And we can include other additional information such as sex, age, ethnicity, and many more. And then developing AI computational methods that leverage this comprehensive genomic data to extract the new knowledge. And the desired output will be a ranked list of variants ordered from the most likely to cause the phenotypes to the least likely one. And the most likely means here that these variants have a higher probability or higher likelihood of being the causative factors behind the phenotypes compared to the other variants on the list. And these factors determined by considering a lot of factors that I will introduce later in this presentation. And obviously, there are several computational methods that have been developed to address this problem. And these computational methods broadly categorize into two different categories: genotype-based method and the phenotype-based method. For the first type, the genotype-based methods, incorporating only the genomic informations to prioritize the variants. However, such methods suffer from some limitation. First, they do not use the patient's phenotype, and they assess the functionality of the genes only based on the functionality of the genes only, without the potential of causing the disease phenotypes, in which that the typical healthy individual might carry a loss-of-function variants in about 20 genes, but without showing the disease phenotypes, which would contribute to the difficulty of interpreting the phenotypic effects. To address this limitation, a new group of research emerged to incorporate the patient's clinical phenotypes into the interpretation of their sequencing data, and based on the prior knowledge, as well as the knowledge that are existing in the databases such as OMIM database. However, such methods still have some limitation, in which that some genes may have not been observed in relation to some phenotypes. And even if such observations exist, they might not be reported in the databases, so leading to a lack of knowledge about the gene information. And to overcome this limitation, a new group of research emerged to incorporate the mouse and model organisms. Although they demonstrated some success, still there are some limitations. So the limitations of the current state-of-the-art approach summarize as follows. First, we face the limitation of the incomplete knowledge about the human phenotypes, and using the mouse model organism and increase our knowledge about these phenotypes, still not all the genes have human orthologs in mouse. And recent studies showed that the more distant model organisms do not easily reproduce to the human phenotypes, and consider that we have a large number of genes in the human, so most of these methods miss large number of gene information. So these challenges emphasize in the ongoing challenges of identifying fully understanding the genes that underlying the disease phenotypes, and then also to incorporate more additional information for the genotype to phenotype association. So our main hypothesis is that there are several useful pieces of information available that we can utilize for the gene phenotypes associations to overcome this limitation. Specifically, we have the gene function, in which that the genes with the specific functions are directly associated with the related phenotypes. So for example, that the genes involved in the insulin metabolism in the Go ontology might be linked to the abnormal insulin metabolism using human phenotypes ontology. Next, we look into the anatomical site of expression or cell types of gene expression, in which that specific tissues or the cell types where the genes are expressed play a crucial role in determining their functional impact. So the genes that are expressed in specific tissues are more likely to be involved in the phenotypes that are directly affecting these tissues. So for example, the genes that are expressed in the neurons of the brain could be directly associated to the neurological or cognitive phenotypes. Lastly, we consider the biological pathways, in which that the genes are usually overconnected through the molecular pathways. So the genes that involved in the same biological pathways often contributed to the related diseases. So by including these different factors, we are be able to more precisely identify the genetic cause underlying the diseases, and also enhance our ability to link the gene to their phenotypes. So our main researchers aim to develop computational methods that leverage this comprehensive genomic data to extract the new knowledge, and the desired output is to have better predictions for the genes and also the causative variants underlying this set of phenotypes. Specifically, this research focuses on exploiting the gene disease information, that including the gene function, the expression patterns, and the pathway involvements. So by integrating these diverse data types, we aim to overcome the current limitation on the variant analysis and significantly improve the gene disease association prediction. From this, we answered the following 4 main research questions. First, how we can effectively integrate the relevant knowledge and leverage the data principle for knowledge representation and reasoning. Second, can we define formal methods that embed the knowledge from these ontologies and knowledge graph into a deep learning models that we can use for variant prioritization or gene prioritization? And how we can measure the effectiveness of such methods in more realistic cases? And finally, can these phenotype-based methods utilizing both genomic and clinical data enhance the diagnostic accuracy of individuals with rare and complex diseases better than other state-of-the-art methods? And to answer these questions, I will be presenting now our computational methods for prioritizing the causative gene and variants. We have developed 3 key approaches or 3 key methods. DLTVIC, our core method for predicting the candidate genes based on the phenotypes, function, and expression sites. EmbedBVB and DeepSVB are 2 applications of these approaches, focusing on variant prioritization. So let's start with DLTVIC, which is our primary methods that integrate various knowledge from different sources. First, we have utilizing the human phenotypes ontology from Human Phenotype Ontology database. Second, we have the mouse genome that incorporates the mouse phenotype databases using the MP ontology. Next, we have the gene product functions that describe the molecular function, cellular component, and biological processes. We also have the cell type specific gene expression that utilizing a single cell RNA sequencing studies. We also incorporate the expression patterns of the genes across different anatomical sites from overall ontology. And all this information is available in forms of ontology. So we integrate it through the cross-species ontologies using PhenomeNet or UPheno that facilitates the semantic integrations of phenotypic information across multiple model organisms. So in addition to that, we incorporate the gene disease association, existing gene disease association, as well as protein-protein interaction network. So all this information available in forms of databases, which required some pre-processing and analysis. Then I integrated them through a biological knowledge graph, where the nodes represent the genes, variants, or the diseases, or any ontology annotations. And we can easily expand this knowledge graph to include more biological information data. So the key idea of DL2FIC method is to employ a feature learning to extract the semantic of the nodes from the ontology and the biological graph. Specifically, it's leveraged the integrated data, including the model organism, phenotype as well as the ontology hierarchy, and to propagate this information through the protein-protein interaction network. So in this process, the new logical axioms features are inferred using Hermit or reasoner, which is supporting the semantic integrations by capturing the semantics of the nodes and their biological interactions to infer the new associations that does not exist previously in the graph. So for example, we can infer this associations through the automated reasoner, although that this association is not existing in our original graph. More formally, DL2FIC methods convert these knowledge axioms into a graph representation by applying a certain transformation rule. So the graph representation involves the conversions of these axioms into triples, where these arbitrary classes, names, and relation names are converted into edge that's representing the relationship between these two different classes. So for example, here feeding behavior, subclass of behavior, would be represented as a triple feeding behavior, subclass behavior, and then will be added these edges into the graph. And this would be enabled us to utilizing the complexity of the axioms into across different species phenotypes ontology. And here is another example in which that in the original graph, FOXP2 has function to a certain class, or Go class, and this Go class is subclass of another class, so we can infer that this genes has functions to this specific Go class. Here is another example in which that class A subclass of two classes R and C. So here a new edges will be created between A and C with relation R and A and P with relation S. So in order to explore the more distant node and exploit the chain of description logic axioms in our knowledge graph, we perform the random walk in the generated graph. And then after the random walk, we run word-to-vec skip graph model, which aim to predict the graph neighborhood patterns. And then once we got the representations for each entity, we can use these feature representation for novel prediction and association. So here is just the general workflow of our methods. First, we have collected the data from different sources and apply our transformation rules to convert this data into a graph representation. And from this graph, we perform several random walks to generate a sentences or corpus. And from this corpus, we run word-to-vec to generate the representations for each embedding for each entity in the graph. But here we are interested in the gene disease entities. So we use this entities to develop a new point-wise learning to rank prediction model that's aimed to predict the gene disease association and also to rank the causative variants. For more detail about this model, how we train it, and how we perform different evaluation, please refer to the published paper. So now we cover DL2FIC. DL2FIC is just one approach for the ontology annotation-based embedding. However, there are many other knowledge-enhanced learning methods for knowledge graph embedding that categorize into three main categories. First, the graph projection-based methods, similar to DL2FIC, which convert the logical axioms into graph triples, and the syntactic methods that generate sentences based on the axioms, and also incorporate some text mining approach, and the model theoretical or the semantic model that represent the axioms directly in the embedding space. So in addition to DL2FIC, we generated embedding using these various approaches and predict the gene disease association using scoring functions. Some of these methods have their own scoring functions to predict the associations between two representations. And all these different approaches implemented through our published work, MAUL library from our group. Okay, so now that we have covered the fundamental concept for embedding approach, so let's explore how we have applied this idea to develop two specific methods for variant prioritization. So I will briefly introduce Embed BVB and Deep SVB, which demonstrate the practical application of the knowledge-enhanced learning techniques. So these two methods have the key similarity that both utilize different background knowledge using different embedding techniques. And the key similarity is that Embed BVB aim to prioritize the single nucleotide variants or the intergenic or the non-coding or coding variants. And for the DeepSVB, aim to prioritize the structural variants. And for both approach, we evaluate the model using the clinical phenotypes and considering variations in their annotation richness, so we can have more realistic setting for real cases studies. So here is the general workflow for the first method. The first part is almost the same for the previous one, to generate the representations for each entity in the graph. So here is the main part, in which that for each variance, we linked to the disease and all the genes in this variant, either one or more than one gene. And for all this gene score, we got the maximum similarity score between each genes and the diseases embedding using the similarity score or the cosine similarity. And after that, we just took the average between this phenotype score and any genotype-based score. In our case, we evaluated CAD score, but we can use alpha sense or any other state-of-the-art approach for the genotype-based scoring. And finally, we assigned this score as a prediction method, prediction score for how likely this particular variant related to the patient's clinical phenotypes. And here is the general workflow for the second model, DeepSVB. Also, the first part is the same. The second part is mainly for collecting features related to the structural variants. We use different features like the structural variants type, GC content, and so on. We apply different features selections methods to see the contributions for these features. And the most important thing that since these structural variants involve large number of genes, we apply and evaluate two different aggregations methods, in which that's either the phenotypes effect due to the single gene effect using the maximum score between the gene and the diseases, or the average, which is considered the cumulative effect for multiple genes in the variants. And after we pre-process these features, we generate another deep learning models train in these features to get the phenotype score for each variant, how they are related to the disease phenotypes. And also for any more details, please refer to our published paper. So we employed different evaluation setting to answer the third research questions, either using the split training evaluation or using the synthetic data testing by inserting the variant into synthetic genome from 1,000 genome. So he will just explain it quickly. We have the list of variants associated with their phenotypes, and we have the synthetic genome collected from the 1,000 genome. So we inserted one variant from this set of variants into this synthetic genome, and then create synthetic patients, and then we run different phenotype-based methods, our approach as well as other state-of-the-art methods. And then we got the ranked list for each variant in this synthetic file. And finally, we will find at which rank this inserted variant is and report the performance at different K. So here is the one of these results shown here for both EmbedBVB on the left and DeepSVB on the right, and demonstrating that our methods outperform the existing phenotype-based tools. So for EmbedBVB, we compared against several methods. However, for DeepSVB, it was the first phenotype-based approach. So we just compared against the genotype-based methods for structural variant prioritization. And in all cases, our approach achieved better than other state-of-the-art methods. There are other more analysis and using other benchmark data sets. So for more comprehensive overview, including these additional tests and analysis, please refer to the published paper. So here's other also related publications. We did some more analysis in collaborating with other Saudi hospitals. So to conclude, our research has made several key contributions. First, we have developed computational methods for phenotype-based gene prioritization that integrating the genomic features, the gene function from the gene ontology, anatomical expression site from Uber-on, the cell type expressions from the cell ontology, as well as the clinical phenotypes. And we leveraged the extensive background knowledge from the ontology annotated databases, as well as the ontology axioms. And we leveraged the knowledge-enhanced machine learning to prioritize the candidate genes. And we addressed different types of variants, the single nucleotide variants, structural variants, non-coding variants. And our evaluations use both synthetic genome as well as the patient-derived clinical genomes that demonstrating the applicability of our approach. And all these methods are publicly available in GitHub. You can access and try them on your own. So before I receive your questions, I would like to acknowledge all the people involved in this work, specifically my PI and now my colleague, Professor Robert Hondorf, for his guidance and support, and our research group, Bio-Ontology Research Group at KAUST, and our collaborators at various universities and institutes. And this concludes my presentation. Thank you all very much for your attention, and I would be happy to address any questions that you may have. Thank you.
Thank you for the talk. I'm not in this area, and I had a question about where some of your data came from.
The data
Okay. For the question.
Okay, I thought you would ask where the data has come from.
Well, not in the sense that you took it from a computer, but part of what you were looking at was expression data within particular tissues. Now, I presume a lot of that might have come from mouse tissues and mapped to human tissue, but I would have thought that in any case, most of it would have come from healthy people and what the normal gene expression was in a healthy tissue. So I'm wondering that when you look at these diseases, do you actually have expression data from tissue from that sick person? Because I could see that having a big impact on any conclusions you're making.
Okay, that's a very nice question, but to be honest with you, we did not look into specific tissues, if it is from the healthy individual or the sick individual. I just download all the gene expression data that's available in this database without looking into their details, if they are normal or not. But
okay, so they weren't related to an individual.
Honestly, no. We just need to collect as much information as we can that might help us to maybe it will not help us directly to predict the causative variants, but it might help to refine our analysis. We can use them as a filter later on, so we can find this expression. Okay, is it helpful for this predictions or not? Then we can ignore it.
Okay, thank you.
But thank you for your question.
Hi, thank you very much. That was a beautiful talk.
Thank you.
Just a bit, I'm still a bit confused on how this tool is used in actual practical form, like on the data. So are we looking at, for example, like a WGS study in which we have patients affected by a particular disease, and we just literally feed the VCF file to your prediction tool?
Yes.
Or do we need to know first candidate variants and your tool shows potential causal association?
Okay. Thank you for your questions. Basically, in order to run this tool, we just need two information: the clinical phenotype in HBO and the VCF file.
So you need controls too?
Just the VCF file and the phenotypes. That's it.
Okay, but the database then needs to contain controls to
Yes.
compare cases between controls.
Yes.
Lovely. Okay.
Yeah, but the VCF file, we will first annotate it with different database, and we will filter the one that less than one math filter for 1%. This is what we did in the Saudi hospital that we work in collaboration with. So they provide us the DNA sequence. We perform the workflow to generate the VCF file. Then we filter these VCF files to have the final, rare variants that we can run our tools in them. And then finally, we run our tools using this filtered VCF file as well as the clinical phenotypes assigned by clinician, and then see where's the scoring ranking variants, and then send this reported variant with the score to our clinician, and then they can study them further to evaluate their effectiveness. And we solved one cases. For deep SPB, we solved one clinical cases, and we have this case and mentioned in the paper.
Lovely. Thank you.
Welcome.